Hey everybody, my name is Mason Hansey. I'll be covering lysogenic conversion. So a little bit of an overview here. The lysogenic conversion overview. Lysogeny is defined as a modified replication cycle in which infected host cells grow and reproduce normally for many generations before they lyse. This lysogenic cycle, a method of viral replication of DNA by the use of the host cell. One of the two ways in which viruses undergo DNA replication, and it involves the use of viral attachment to introduce the viral DNA into a host cell, also known as lysogeny, as I guess I stated earlier. The lysogenic cycle steps. Step 1. The viral DNA is introduced to the cell but doesn't destroy the host cell DNA. It stays inactive and it is called a prophage. Step 2, it stays inactive and codes the protein to suppress the prophage genes. Step 3, the fusing of two deep pieces of DNA and with replication of a normal cell, the viral cell replicates it. Step 4. This is the step in which lysogenic conversion happens. This is the result of changing a relatively harmless bacterium into a pathogen by changing the phenotype. Later, the excision of a prophage is called induction. So it is obviously here that you can see that it completely kind of changes the makeup of that bacterium cell into a almost like a pathogen of sorts that could spread the virus. Steps continued. 5. Induction occurs. 6. The lysis occurs. 7 is the assembly of the viruses. 8. The cell is full of virions and bursts, causing the virions to spread further. This spreads that virus further throughout the cell. 2 examples of lysogenic conversion are rheumatic fever and diphtheria. That is from the book. Difference between lytic and the lysogenic cycle. One difference between the lytic and lysogenic cycle is the fact that in the lysogenic cycle, the virus integrates itself into the DNA. The lytic cycle doesn't do that per se. In the lytic cycle, the actual replication of the virus takes place separate the actual host DNA. This is opposite of the lysogenic, where the replication of the virus takes place with it. And another difference is the fact that in the lytic cycle, the whole cell is completely overtook and doesn't retain mechanical properties separate the virus. So really in that lytic cycle, it is that bacterial cell has no control. It can't go about its functions. The lytic cycle completely takes the whole thing over. So yes, these are my references. Thank you and have a good one.